Start the recording and I'll hand it off to the first tutor.
Alrighty, hi guys. Okay, so today I'll be introducing the concepts of reaction rates, rate laws, and reaction order. So first starting with the rate of a reaction. The rate of a reaction describes how fast the concentration of reactant changes to product over time. So let's look at an example reaction. So if we have A plus B goes to C plus D, where lowercase letters represent the stoichiometric coefficients or the number of molecules of each reactant or product, and uppercase letters represent the reactants and products. If we have a reaction that looks like this, then the reaction rate would be given by this equation. So the rate equation is defined so that it doesn't matter which species out of A, B, C, or D you follow. They are all like included in the rate definition. So really the rate of the reaction is describing how fast the reactants disappear and also how fast the products appear. And this is reflected in the signs. So there's negative, if you notice, there are negative signs in front of A and B, and there are positives, it's just positive in front of the expressions for C and D. And hopefully this makes intuitive sense because A and B are the reactants, and so they would be disappearing in a reaction, and so the change of their concentrations would be negative. Whereas for the products, they are increasing over time, and so the change in their concentrations would necessarily be positive. So each expression, each derivative is also divided by the stoichiometric coefficient. And this is because the stoichiometry of the reaction affects the reaction rate. So to see this, think about A goes to B versus A goes to 2B. So one molecule of A goes to one molecule of B versus one molecule of A goes to two molecules of B. And it makes sense that in the second reaction, the concentration of B increases twice as rapidly as the first reaction. So that's why we have to include stoichiometric coefficients in our definition of reaction rate, because the number of molecules of each reactant or product affects how fast the reaction progresses. So the rate also depends on the concentration of the reactants, because reactants have to come close together and collide in order to react. So the higher the concentration of your reactants, the faster they can find each other in solution, and the faster they can collide, and then therefore the faster the reaction. So like, let's say that we have a mixture of red and blue molecules. Blue they're calling A, and red they're calling B. So if you're thinking about the space in a reaction container in terms of a grid, and if we say that a collision happens when the two reactants occupy two adjacent boxes in the grid, then the probability of this happening, of a collision happening, is PA times PB. And PA and PB here is equal to the total number of molecules of A or B over the total number of grid boxes. So So this is basically showing that the collision rate is proportional to the product of the concentrations of the reactants. So the higher, again, the higher the concentration of your reactants, the higher the reaction rate. So how can we incorporate that into the reaction rate definition? So we need to include a term in our definition that takes this into account. And then once we do that, we get the rate law, which basically it describes the rate of change of concentration with time. And it's defining the relationship between reaction rates and the concentrations of the reactants. So let's consider a simple elementary reaction. And by the way, an elementary reaction is basically just a reaction that goes from reactants to products in one step, as opposed to multiple steps. But so if we have an elementary reaction, A plus B goes to C, then the rate law would be rate equals negative dA over dt equals dC over dt equals K times the concentration of A times the concentration of B. And here K is the rate constant, which reflects the frequency of collisions and the fraction of collisions that actually lead to product formation. So K is our term that takes into account the concentrations of the reactants, and therefore it takes into account how fast collisions occur based on what we said before. And it also takes into account which of these collisions are actually productive, because you can imagine that just because two reactants collide doesn't mean that they necessarily lead to product formation. So K is also telling you what percentage of collisions is successful. And I think in this section, you'll be learning that reactants need to be in the right conformation, and they also need to have a sufficient amount of energy in order for the reaction to occur. So those things are important. And also, if a reaction uses a catalyst, which is any substance that increases the reaction rate without being consumed itself, the catalyst has to be included in the rate law, even though it's not in the overall reaction, because its concentration greatly affects the rate. The more catalyst you have, the higher the rate. So up here is the reaction where you have two.
I'll continue talking.
Oh. Did somebody have a question?
Sorry, it was me. Sorry. No.
Oh, Lisa. Okay. So here you have a reaction where you have two reactants, A and B. They collide, and then they form the product C. So this is what the corresponding rate law looks like for that. And then here you see a reaction with one reactant and one catalyst, which forms a catalytic complex, and eventually you form C from A. So the rate law has the concentration of the catalyst included, because as we said, the concentration of the catalyst would affect the reaction rate. The more catalyst you have, the faster the reaction. So the reaction order is defined as the sum of exponents of the concentrations in a rate law. And in other words, this is basically the number of molecules colliding, since the exponents in a rate law represent the stoichiometric coefficients that are required for each species. And then the units of the rate constant K depend on the reaction order. So K will be different, or the units of K will be different depending on how many molecules of each reactant are required. So first, let's look at reactions that have only one reactant. These are called unimolecular reactions, and they're called first order because the sum of the coefficients equal 1. So this is the general form. A goes to C, and then this is what the rate law would look like. dA over dt. So we're only following dA over dt. We could follow dC over dt. In that case, there wouldn't be a negative sign here, but usually we would follow like the change in the concentration of a reactant, in this case A, but it doesn't matter because it'll be the same, just might be different signs. But this is the rate law of a first order reaction. Oh, and the units of K for a first order reaction would be S to the negative first power. And this is because the units of concentration would be molar concentration, which is denoted by capital M. And then, so on the left side would be capital M over seconds. So to have the right side of this equation equal the left side, you would have to have S to the negative first power for K to have M over S on this side as well. So for second order or bimolecular reactions have two reactants, and these are called second order because the sum of the coefficients in the rate law equal to 2. So this, you can think about it as, here you can think about it as A to the first power, B to the first power. So 1 plus 1 equals 2. That's where second order comes from. And then, yeah, so this is what the rate law looks like. This time, because there are two reactants, both of their concentrations affect the rate of the reaction. And if you solve for the units of K, you would get capital M to the negative first power times S to the negative first power. This is different from the units of K for first order. As I said, the units of K depends on the reaction order. So they will be different for each kind of reaction. And I encourage you guys to try and solve for K on your own to see if you get the same thing. And then for zero order reactions, this is basically the same as a unimolecular, except that it has a catalyst to aid the reaction. And then, so for a zero order reaction, the rate is actually independent of the concentration of reactant. And this happens when the reaction rate depends on the catalyst, and the concentration of the catalyst is a lot lower than the concentration of the reactant. So like this is represented here in like this thing that I drew. So you have a lot of molecules of reactant A, and then you only have a very small amount of the catalyst as such. And so no matter how much of A you add, the reaction rate will not increase. So that is why the rate law does not have the concentration of A in it. So also the units of K is, in this time it's M times S, or basically M over S. And try to solve that on your own too. And what is the use of these rate laws? We can use these rate laws to find what is called the time dependence of concentrations. Basically, we can know the concentration of a reactant at a given time t if we integrate the rate equation. So experimentally, we can manually do this. We can measure the concentration over time. And we can do this by measuring the absorption of light, which changes as the concentration changes. So I won't go into the details of this, but basically this is how experimentally we can measure a concentration of a reaction over time. But then we can also integrate the rate equations to get expressions that give us concentration over time. And then I'll be integrating the rate laws for zero order and first order, starting with zero order. If you recall the rate law, we had dA over dt equals negative k. So if you integrate both sides using limits A sub 0, which is the initial concentration of A, at time 0 to concentration of A at an arbitrary time point, you would get this. And then if you integrated that, you would get this equation. This basically tells you that the reactant disappears linearly with time because negative k times t is in the linear form. So yeah, so this tells you that the reactant will disappear linearly with time. And we can verify this experimentally. So if we measure the reactant, concentration of the reactant over time, the graph will be linear, like we said. And so that's just, a sanity check and that they match. And then, whereas for a first order reaction, the reactant disappears exponentially. So if we do the same thing for the rate law of the first order reaction, and if we rearrange the rate law, we get dA over A equals negative k dt. And then if we integrate that, we get this equation. And then if we take the exponential on each side and then isolate A to get an expression for A at time t, we get this equation. And that tells you that the reactant disappears exponentially with time. And we can verify this experimentally. So, and then if we graph that out, we get something that looks like an exponential function. So that also checks out. And then, yeah, that is all I have, and I think I'm running out of time. So I will stop sharing and let the next hear.
Hello, everyone. Okay. All right. Okay, so this is the goal we are going to talk about today. There's a ton, and I think the third topic is way more interesting, so I will try my best to run through all of them. Okay, so I'll assume that you have some decent amount of knowledge of zero order and first order reaction. So some terminologies I want to clarify a little bit is reaction order is just the sum of number of molecules participating in an elementary reaction. So zero order is not an elementary reaction, so we don't consider that. For first order, it's the only one molecule. Second order, and it's a bimolecular reaction, which is very straightforward, I think. The rate law is the differential equation of change in concentration with respect to time. So usually write it as the concentration over dt. That thing is what we call the rate law. And we also have this integrated rate law, which is the integration of rate law that gives us the concentration as a function of time, which is a little bit more intuitive to look at. Okay, so so the way we write the second order rate law and the first is very similar to we write the zero order and first order rate law. This is just a chart that you can maybe if you want to refer to, but it's just basic stuff. So so the way we write second order rate law, okay, so second order reaction is a bimolecular reaction. So let's consider the first case where we have only one type of reactants. That means we have two molecules of the same type, right? So A and A they collide with each other. So let's write our rate law as negative K times concentration of A times concentration of A, and let's separate our variables. I think you did that yesterday, so just basic integration stuff. Separate variable and integrate from A naught to some arbitrary concentration, and so integrate with respect to time from time equal to 0 to some arbitrary time, and that gives us this expression. With a little bit of rearrangement, we get this expression. So that's the law for second order reaction if we only have one type of reactants. Done, very simple. So let's make the situation a little bit more complex, considering two types of reactants. So we have A and B, two molecules. Rate law is easy to write, right? So just here you can see. So let's now consider, what if I have the initial concentration of two kinds? They start from same concentration, so A naught equal to B naught, and we know the reaction is that we will one molecule of A collide with one molecule of B. So if we start from the same numbers of molecule of A and B, and because of the stoichiometry, you will end up with the same concentration of A and B all the time as the reaction proceeds. So it basically just returned to the same case as we have just now. So then that makes the math very easy, just essentially the same thing. Okay, but what if we have a non-equal concentration to start with? Then we'll have to do the integration of this expression. The pain here is because we have two terms that's dependent on time. So one trick you can consider is to write B in terms of A. We will take the advantage of the stoichiometry equivalency of conversion of A and B, because meaning the changing A equals to changing B. So that enables us to write a concentration of B in terms of concentration of A and plus some constant. And then we can plug that in and do some integration tricks I'm not familiar with anymore. And then we can eventually reach this expression. The detail of the integration is not important here, I think. I don't. Okay, so let's look at this messy expression. Let's now consider if the concentration we start with is not comparable to each other. For example, if we have the amount of A greatly exceed the amount of B. And what that means is that as reaction proceeds forward, the concentration of A is not going to change significantly because it starts from a whole bunch, and then it's just essentially this concentration term will keeps unchanged during the reactions. So then we can replace this term by the initial concentration, because we say it's not going to change that much. That enables us to combine these two terms into a new constant K prime. Why do we want to do that? Because it essentially turned this expression into something very similar to what we see in the first order rate law. And that makes integration easy. We can just write essentially the same thing. We integrated from the first rate law and using a different rate constant K, which is the product of K times A naught. And these two things, they are all constant. This is a very important case. I want to remind you, well, this K prime term is what we call the pseudo first order rate constant. It's very biologically relevant. You will see it shows up a lot in ligand protein binding example that I hopefully to go over today, but maybe not. Okay. The other case that's more nasty is that you have comparable starting concentration of A naught and B naught. And then your textbook say it's too complex to analyze. So we should not talk about this too much here today. Okay, so another basic tool that we want to use is half-life. Half-life is a very useful parameter to show you how fast a reaction proceeds, but it is the amount of time required for initial concentration of reactants to decrease by 50%. It's not hard at all to figure out half-life. You essentially just set your concentration to half of the initial concentration and plug back into the integrated rate law and solve for T. And if you forget, I have this integrated rate law for 0, first, second order, or written out, you can refer to. Okay. Yeah, so what I want to say is that the half-life is just a special point. It's just a special point on the integrated rate law graph, which is the concentration versus time graph. So it is the point corresponding to half of initial concentration. So it's not something new, but it's kind of handy to just give you a direct readout of the speed of a reaction. But I do want to remind you the dependency of half-life on concentration for zeros, first and second order, they're all different. And actually they're so different. If for zeros order, when the initial concentration increases, your half-life will also increase. And it makes sense because the rate for a zeroth order reaction is a constant. So if you start with more reactants, it makes sense the time required to reach 1/2 of that amount will also increase. First order, the half-life of first order is very famous. You should all remember that. And it's initial concentration independent. The half-life, the interesting thing for second order reaction is if your initial concentration increase, your half-life will essentially decrease. Why is that? Because if you have a higher concentration, it means it will easier for a collision to happen. So it essentially makes the reaction faster. So you can also see something that's kind of intuitive is higher concentration could actually impede a reaction. Okay. Another interesting thing is that if you fixed A naught the same, which is easy to do, right? You will see that first order and second order, they behave actually kind of similar from the beginning, but second order will start to slow down. The decaying for second order start to slow down as the reaction proceeds. And it's basically because of we have this square term for second order reaction. That means as your reactant concentration goes down, the rate will slow down more dramatically than a first order reactions. And I think that's the major difference you can see from this graph. Okay. Other things, I don't think that's too important. And we just went over this last tutor. Okay. Then let's now make our system a little bit more sophisticated by considering two sequential step of reactions. So we have A converted to B and B converted to C. We are still not considering reversible reactions, but in this system, we want to ask a question that is that the question we want to ask is that what influence will bring to the final product C if we have different rate constant Ka and Kb. So a terminology thing is intermediate. It refers to something that's produced in the process of a reaction and completely consumed during the reaction. So it will not show up in the overall reaction. Okay. So we want to, so as I said, we want to study C. So we start by writing out the rate law for C. Writing out correct rate law is very important, and I encourage you to try out that by yourself. So yeah, hopefully you can understand this. So it shows up as a first order reaction, but so we want to know what is how can we write this term? This term, as you remember, we can write it as a function of time, which we can get from integrating rate law of B. So let's write the rate law of B, which is not super straightforward to write because B is being produced while it is also being consumed. So I remind you, be very careful at this step. So this term plus Ka times the concentration of A means B is made from A, and minus K2 times concentration of B means B is being consumed. If you don't understand why I write this term as B but not concentration of C, please put it in the chat and I will reply to. So we have this expression, but something here shows up, tells us we need to further break this term down to the concentration of A as a function of time. So then we write the rate law of A, which is a first order reaction, and we note the integrated law of first order reaction. Okay, finally, we can plug that expression into this term, and we get here. And then we do the integration, and that gives us this expression. Okay, so now what? Now we have this B as a function of T. We can plug that in, we can plug that to this master expression of which is the rate law of C. Okay. Then we have a rate law of C and let's do the integration on this rate law. And that gives us this expression. You should not remember this at all. I've never used this in my entire life, but we can still appreciate this. Why? Because this graph is also what you saw from the lecture. It's basically saying no matter the order of slow step and fast step, the buildup of C will essentially be the same. Why is that? Because you can see from this expression, it is determined by this difference of two rate constant. As t proceed to infinity, these two terms basically become 0. So this term basically determines how much of C will build up. Okay, but without doing any math, I want you to remember this very important concept of rate limiting step, which is the slow step in a reaction will determine the overall kinetics of the reaction. Yes. Okay. Slow step dominance. That's what I want to say. Okay. I'm running out of time, but so steady state. Steady state is a state where your concentration of product and the concentration of reactants does not change much over significant period of time, despite your reaction still going on. So I want to remind you, steady state is not exactly the same thing as equilibrium if you consider homeostasis. So cell is almost never at equilibrium. Equilibrium is defined by delta G equals to 0. We talked about last week. Cell is almost never at equilibrium, or you would be crazy. But the metabolites usually doesn't fluctuate largely over time. Okay, that's basically all I want to say. I hope you can go through the derivation of this expression because I'm running out of time. And this is a very cool analogy to understand steady state kinetics. And why it is important, because the metabolic concentration of steady states is metabolic reaction is extremely sensitive to intermediate concentration in a steady state. You can understand that by a fractional bond and how would that influence enzyme activity, allosteric regulation, and so on. Also reversible reaction. So reversible reaction is that you have to consider an extra term denoting the rate of reaction going backwards. But the duration is the same as what I just went through. So you write out the rate law first and then you do the integration. That will give you this expression. Okay, so things to notice is that the reactant will not decrease to 0, but decrease to some constant value of A, some constant value. Okay, and if you went through this part, you will find the very interesting relationship between kinetics and thermodynamics given by this ratio of forward and backward reactions, and it has very important implications in how you think about ligand protein binding reactions. Okay, and yeah, I hope you can look at these notes. This example is something Professor Korean is studying, so I encourage you to look at it. Okay, and I have a practice problem, which is just writing out the rate law, and you can try it out. Okay, that's all I had today. Thank you.
All right, let me share my screen. All right. All right, so for this last presentation, I'll be going a bit more detail of what affects the reaction rate and how they all come together, all these factors that affect the rate of reactions, how they all come together in the Arrhenius equation. So let's get started. So throughout this past hour, you've heard the term rate constant a lot, but in reality, even though it's a constant, this rate constant, it's only constant when changes in concentration are under a set defined set of reaction conditions. So for example, constant temperature, constant medium. But when these reaction conditions are varied, this rate constant is no longer uh, a constant. So I'm going to be going to different factors that influence the rate of a reaction. And some other thing that's important to note as I go along this presentation are the concept of catalysts. You've all heard of them before. Enzymes are very important biological catalysts, and they speed up a reaction, the rate of a reaction, but do not appear in the overall balanced reaction. And that's because they're not consumed. So for example, if the net reaction is A plus B producing C, if you actually dig a bit deeper and look at the actual reaction, D is the catalyst. So first A reacts with D to produce an intermediate E, and then E reacts with B to produce the product, and the enzyme or the catalyst is also produced. So when you combine these two together, you get this net reaction. But this catalyst D is very important because you can imagine that if A were to react with B directly, this K will be very small, so it's not possible. And what this catalyst does is that it lowers the overall reaction rate and makes it possible for A to react with B. So without further ado, let's get on to the certain conditions that affect the rate of a reaction. The first thing that Wu actually brought up in the first presentation was for the reaction to proceed, the two reactants have to actually collide with each other and come together for the product to be produced. And the rate at which, for example, in this case, A and B come together is defined by the medium which they're in and the speed they're moving in. And the fastest possible speed that A can come to B is limited by their rate of diffusion. And diffusion is essentially when the two molecules are freely moving in space. But you can imagine that this is not actually happening in a cell because a cell is very crowded. There's a lot of other things going on. So in reality, reactions are happening much slower than this maximum possible rate, which is limited only by diffusion. But ideally, in water, this diffusion limited rate of collision is 10 to the 10 per molar per second. And to model this, the fact that the rate of a reaction depends on the reactants colliding, we can use this equation, which is simply the rate of collision depends on K collision and the concentrations of A and B. And it makes pretty intuitive sense that if you increase the amount of the reactants, they're more likely going to collide. So that's why the N collision increases. But as I said, most reactions aren't limited by diffusion and they're much slower. So why is that? Well, one reason that these reactions are actually slower and are not limited by diffusion is the fact that the reactants have to come together in a specific orientation. So it's not just if A and B come together, the product is produced. They have to come together and collide in specific parts for A and B to actually produce C. So to take this into account, we can multiply the K collision, which can be diffusion limited, by the fraction of the collisions that actually lead to a product. And we get this final term, which is we call the pre-exponential constant, which will make sense later why it's called a pre-exponential constant. And just as an example of the fact that these reactants have to come together in a specific orientation is a pretty important example, which is ATP, which is the energy source of a cell. And for an enzyme to actually use ATP, it has to collide to the business end of ATP, which is this triphosphate group at the very end. For example, if an enzyme was to attack the adenine group or the ribose here, nothing would happen. So the enzyme has to collide and react with this ATP phosphate group for something to happen. We can use this equation to take this into account. And another reason why reactions are actually, not all reactions are productive is the fact that the collision has to provide enough energy for it to overcome the activation energy. And the activation energy is the minimum energy required to bring the reactants sufficiently close enough to react together. And another way to put this is that the activation energy is a difference in energy between the reactants and the transition state. And this transition state is, you can think of it as the highest energy point in a reaction coordinate. And going back to the example I brought up in the previous slide, and when you want to use ATP as an energy source, you can attack, for example, if the ATP is in a water environment, the water has to attack the terminal phosphate group and it forms this intermediate. And this is the highest energy point along this reaction. And you can use this symbol, the dagger symbol, to illustrate that this is a transition state. And this goes at this peak in this reaction coordinate graph. And if the reaction was to overcome this high energy point, it can produce the products. So how can we take this into account when we're deciding the rate of a reaction? Because that's the ultimate goal, to bring all these different factors into play to get a more accurate picture of what the rate constant is. And this is where we can use a concept that's been extensively used so far in this class, and it's the Boltzmann distribution. We can incorporate this Boltzmann factor, which shows where the molecules are in an energy diagram, into our rate constant to signify that we need to have this minimum EA, or the activation energy, for it to actually produce the products. And note that this is exponential, which signifies how important it is for the reactants to have enough energy to overcome this hump in the reaction. So now I've talked about orientation, the fact that they must come together in a specific form, temperature, the activation energy, and then the fact that the medium also affects the rate of diffusion. So how can we bring all this together? So we can, instead of writing K as a general vague concept of the rate constant, we can substitute this term here, A times this Boltzmann factor. And now it makes sense why we called A the pre-exponential constant, because it comes before the exponential term. And we can use this and plug this into the, for example, a second order reaction instead of K. And this is essentially saying that the rate of a reaction depends on all these other factors that influence this rate constant. And the Arrhenius equation takes a natural log of this term here, and actually this equation here, because it makes it easier to plot on a graph, and it shows a linear relationship between the logarithm of the rate constant, the natural logarithm of the rate constant, and the inverse of temperature. So why is this useful? Well, there's several applications of this Arrhenius equation. For one, it shows the temperature dependence of the rate constant. And that makes sense. As you can see here, as you increase the temperature, the rate constant also increases, because if you go back to the Boltzmann distribution, the higher temperature you have, the more likely you'll see particles in higher energy states. And that's why, and they're more likely to overcome the activation energy required to produce the product, for example, C. And that's one useful application of the Arrhenius equation. Another very useful application is that if you notice here, this is essentially the general linear equation, which is y equals mx plus b. So if you plot the natural log of the rate constant in terms of 1 over t, which is our domain, we can say that the slope of this linear graph is simply negative of the activation energy over the gas constant. And this is a simply constant. So by looking at the slope, we can determine what the activation energy is. But not only this, the y-intercept is the natural log of A. And by doing so, by figuring out what A is, we can determine what fraction of the collisions are actually productive, because recall that A is simply K collision times the fraction of productive collisions. So again, this is a very useful equation in determining the various factors that govern a reaction. And then finally, I briefly talked about this. The idea that a reaction has to overcome a transition state is called transition state theory, and that it's simply that the transition state is the highest energy point in the reaction trajectory from getting the reactants to the products. And the theory assumes that the unreacted reactants are in equilibrium with the transition states. And if you recall, I talked about catalysts briefly in the very first slide. So how can catalysts speed up a reaction? Well, based on our discussion so far on the various things that influence a reaction, for one, they can lower the activation energy, which if you recall, I said that there is exponential dependence on this activation energy. So you can imagine lowering the exponential term will have a significant impact on the rate of reaction. Another thing that enzymes can do is actually increase the pre-exponential coefficient A, and this can be done in several ways, or it can do both things, which is just better, which will also increase the reaction even more. And just as you can see in this graph, the catalyst can lower the activation energy and increase the rate of a reaction without being consumed. And how they can lower the activation energy, for one, they can form favorable interactions with the transition state. Like for example, with the ATP example, they can form favorable bonds with the negative charges on the phosphate groups, and that will lower the transition state by stabilizing it. Or to increase A, they can confine the reactants to a smaller space. So that's why and if it makes sense, if they're confined to a smaller space, they're more likely going to collide, and that will also increase the rate of a reaction. And yeah, that's all I have. And yeah, I went over